real-time feeding, and this is experiment condition. So you have a free mouse and then free piezo-1 viral injection, and then we wait some time to express the ion channel, and then we inject the M2 core and do the MG stimulation. And MG stimulation is about same as the one before, 0.5 hertz, and then total 5 minutes. So this is a same mouse. This is the field zone, and this is a magnetic field is off, and so it's just roaming around. The video is somehow doesn't work. I don't know. There is some interference with my PowerPoint presentation and the video or internet broadcasting. I don't know why, but it doesn't work. So, but it quite focused and into food just roaming around. And so this is a cumulative duration time, and then it is increased about in least 3 times higher, and then the DAPI C4 shows that clearly these neuron are activated by the mechanosensitive ion channel stimulation. So that's quite exciting to us. And then we extended this study into social novelty study with multiple animal studies. So here you have 2 mice, and there is M1, M2, and M2 is a novel, and M1 is familiar. There is a mouse in each cage, and then this is a control experiment, and it's better. They just roam around. There are 2 mice, and then just go to M2 and also M1. This is the mouse, the track is shown, so it goes almost there. So with the magnetic stimulation, the video doesn't really work. So as you can clearly see that they spend most of the time with M2, and this is because of the VGAS stimulation. When you do the VGAS stimulation, they increase the social behavior, and then it go into M2 region. So this is our study with a future. We can clearly see that more interaction time into M2 region. And this is a study by optogenetics, and it's comparable. And one clear distinction is that we utilize 2 mice, whereas they use only 1 mice. The mouse was optically linked into the system. So, but we could say that because our system is a wireless, and also we could long distance simulation. I think that could be quite exciting. So this is the summary of our system. So you have adenovirus pH1, and then M2 nanoparticle, and the ion channels. I talked about the piezo-1, but you could utilize any mechanism of ion channel, and you could patch to adenovirus length in AAV, and the particle what we used was about 200 nanometer to 500, but you can tune the force if you need 7 piconewton, 10 piconewton, you could do that. You could go down to 2 piconewton and so on. And biocompatibility, and these are non-toxic, non-leaching, meaning no metal ions are leaching because it is surface is well coated with the silica and so on, so it's well protected. And the apparatus is quite easy, and it can have a quite large long distance working area, and you could go into primate and so on in principle, and the channel gating frequency can be tuned. not as good as optogenetics, because you can laser, you could have a ultra fast and an ultra slow and a medium. The point is that this is a wireless energy transfer fourth generation. You could do that, and then it's a bioorthogonal and a reliable kinetics. Then you could do all kinds of molecular cellular specificity with it. So people have done beautiful, amazing studies with optogenetics, and maybe magnetic genetics can be another potential tool for going to maybe more complex or bigger scale object, and that's something that we are interested in. Maybe you can go to multiple targets at once in a large area, going to clinical studies for brain disease. At the same time, we could also go for neuropsychiatry studies and so on. So this could be, maybe complementary. And this slide shows that you all know that magnetic tweezers, optical tweezers, and so on. Yeah.
Yeah, we are over time by 10 minutes.
Oh,
already,
is that right?
so
Okay, so this is our last slide. So just telling you that this working distance is quite important, and we can really show you that force and the distance can be tuned, and we are actually this is we have UI controller and Mac 35 and 6 centimeter. We are interested in, actually, we are looking for neuroscientists who can work together to develop our tool, with a magnetic genetics and so on. If you are interested in, please visit the website, and then we could, communicate more. And with that, I'd like to close my talk, and thank you so much for your attention.
All right, thank you so much, Ibu. So we are absolutely over time. So if you don't mind, we will move the questions to the panel that we will celebrate after all the talks in this first session. And so without further ado, I would like to introduce our next speaker, Shigeki Kiyonaka. Once again, my apologies if I didn't pronounce your name well. It's not easy for us. And so Shigeki is an associate professor at the Nagoya University in Japan. And today he will be talking about chemical approaches for understanding physiological roles of glutamate receptors in neurons. So please, if you can share your screen, remember that you have 25 minutes plus 5 minutes of questions. And whenever you want, you can start. Thank you very much.
Okay, thank you for your introductions. Can you hear me? Is it fine? Okay, that's fine.
Perfect.
My name is Shigeki Kiyonaka from Nagoya University, Japan. I would like to thank the organizer, especially Raphael and Arizol, for giving me this great opportunity to present our researches. Our research direction is understanding brain function by chemical approaches. One research direction is visualizing neurotransmitter receptor by chemical labeling technique. Another direction is functional control of the receptor proteins by chemogenetics approach. First, I talk about fluorescent visualization of neurotransmitter receptor by chemical labeling. In excited neurons, glutamate receptors are highly expressed in the positron side, which are categorized into ion channel types and GPCR types. The ion channel types are further divided into three classes, mainly three classes: kainate, NMDA, and AMPA receptors. Among them, AMPA receptor has essential role in synaptic plasticity, a putative cellular mechanism of our memory. Recent studies have revealed that the number of AMPA receptors is strictly controlling during synaptic plasticity. For example, during long-term potentiation, exocytosis and lateral diffusion of AMPA receptor are facilitated to increase the number of cell surface AMPA receptor on the postsynaptic membrane to enhance postsynaptic current. In contrast, in the long-term depression, endocytosis and degradation pathway are accelerated to decrease cell surface unprecedented. To visualize this event, unprecedented tagged with pH-sensitive GFP variant on the N-terminus have been widely utilized to visualize cell surface unprecedented selectively. However, in most cases, as I can show you, GFP tagged unprecedented are overexpressed in the neurons. As I mentioned, the strict control of the number of unprecedented are essential. However, the number of unprecedented are already changed by overexpression of unprecedented, GFP target unprecedented. Thus, we should visualize endogenous unprecedented. The potential approach would be utilization of antibody. However, considering the narrow size of the synaptic cleft, antibody cannot visualize synaptic unprecedented. Thus, ideally, native unprecedented should be tagged with small fluoropher. To visualize endogenous unprecedented, we focus on chemical labeling technique termed ligand-directed acylimazole, LDI chemistry, developed by Professor Hamachi, my previous boss. In this method, we utilized labeling reagent having the ligand group and fluoropher which are connected via reactive acylimazole linker showing moderate reactivity. After addition of the reagent in the culture medium, the ligand group is recognized to the target proteins. As a result, the fluoropher is transferred to the target protein by covalent bond formation with lysine or serine residue. And the resultant ligand group can be removed by simple medium exchange. To apply this method to unprecedented, we designed labeling reagent for unprecedented terms CAM2 or G, which has selected ligand to unprecedented and organ green as a fluoropher. These are connected via reactive acylimazole. First, the chemical labeling was examined Hec cells transiently expressed with unprecedented. We need several hours for the chemical labeling because of the moderate reactivity of the CAM2 reagent. Thus, the cells are needed to keep at low temperature such as 70 degrees during the labeling period to suppress the internalization of the labeled unprecedented. This confocal image was obtained after adding the labeling reagent to the culture medium. This indicates that labeling reagent does not penetrate into the cells. And the cells are incubated 3 hours at 70 degrees. After the medium exchange, confocal image clearly indicates a strong fluorescent signal from the cell surface. To confirm the selectivity to unprecedented of the labeling, we examined Western blotting using anti-organ green antibody after the chemical labeling. And zinc labeled band was observed corresponding to the molecular weight of unprecedented. Importantly, the labeled band disappeared in the presence of competitive inhibitor. Indicates that selective labeling to cell surface unprecedented under live cell conditions. Then we applied this technique to label unprecedented endogenously expressed in neurons. In these cases, primary cultural neurons were prepared from cerebral cortex. As is the case of the labeling in Hec cells, culture reagent was added to the neuron and incubated 4 hours at 70 degrees. I show here the Western blotting using anti-organ green antibody indicates selective labeling to unprecedented endogenously expressed in neuron with fluoropher. Next, we examined fluorescent visualization of endogenous unprecedented in the cultured neuron. In this experiment, cultured hippocampal neuron were incubated with CAM2 AX488 having RX488 instead of the oligonucleotide under live cell condition. To visualize the shape of neurons, after fixation with paraformaldehyde, dendrites were visualized using anti-MAP2 antibody. As shown here, the green labeled signals were observed along dendrites, as shown here. Importantly, the fluorescent labeled puncture merged well with the anti-PSC95 antibody, indicates that we succeeded in chemical labeling of endogenous AMP receptor highly expressed in the synaptic puncture in neurons. We succeeded in chemical labeling of endogenous AMP receptor by LDI chemistries. However, we have some limitation in the original method. First, this experiment should be conducted at 70 degrees to suppress endocytosis of AMP receptor during the labeling period. Moreover, we need to incubate the neurons with serum-free medium during the labeling. This is because hydrophobic and aromatic chlorophyll is recognized to albumin included in the serum, which cause non-specific labeling. To improve this drawback, we developed a modified method by combining the LDI chemistry with biosonar chemistries. In this method, tissue group click chemistry donor is covalently labeled to AMP receptors. We can conduct chemical labeling even in the serum containing medium at 37 degrees because TCO is relatively small hydrophobic group compared with large chlorophyll of the original method. After tissue labeling, chlorophyll is covalently attached to AMP receptor using TCO and tetragen ligation, one of the fastest quick reaction. Although the labeled AMP receptor are internalized during the first step, cell surface AMP receptor are selectively visualized in the second step. Okay. First, we examined the two-step labeling in hex cells transiently expressed AMP receptors. In this experiment, CAMS TCO having tissue group was incubated at 37 degrees. In this experiment, CAMS TCO group was incubated at 37 degrees and added tetragen probe for 5 minutes. The labeling was analyzed by Western blotting, as shown here. In the CAMS tissue labeling, AMP receptor was selectively labeled even in the presence of serum. Importantly, the label in the original method at 17 degrees, in the original method means direct labeling method. Confocal imaging, this image shows that the labeled AMP receptor were internalized as shown here. In contrast, in the current two-step labeling method, labeled AMP receptor are selectively visualized on the cell surface. Another advantage of the two-step labeling method is that tetragen and chlorophyll show high fluorescence after tetragen tissue ligation. Tetragen group act as a fluorescent quencher, and this quenching effect is significantly weakened after reaction of tetragen and TCO. Thanks to the turn-on type fluorescent properties, we can estimate the reaction kinetics of the second chlorophyll labeling on the cell surface by adding the tetragen probe in the culture medium, which determined chlorophyll labeling was finished within 3 minutes. Then we applied this method for visualizing cell surface AMP receptor in neurons. In this experiment, CAMS TCO was incubated for hours at 37 degrees. Confocal imaging after immunostaining clearly indicates that endogenous AMP receptor were selectively labeled in neurons. In this case, tetragen Alexa 488 was added for 5 minutes. Another advantage of the two-step labeling is that we can easily exchange the labeled chlorophyll in this method, and we label CETAW 647 using tetragen CETAW 647 instead of the tetragen Alexa 488. This probe, CETAW 647, is highly stable and show unique fluorescent lifetime. Thus, using the fluorescent lifetime imaging microscopy, we can extract Z647 component from the fluorescent image, which allow quantification of cell surface amplitudes by subtracting the background signals. This reveals that the ratio of amplitude in dendrite and spine was 3:10. Next, we analyze the lifetime of endogenous amplitude in neurons using two-step leveling method with biochemical analysis. In this experiment, the cortical neuron were incubated with CAM2TCO for 10 hours at 37 degrees, and the neuron were incubated for each period, such as 0 to 4 to 120 hours. After the incubation, the remained surface amplitude were labeled with tetragen FL. Then analyze FL labeled amplitude by Western blotting. I show here the half-life of cell surface amplitude was 33 hours in neurons, okay? We also determine the half-life of amplitude in HEC2 CT cells in the similar method. Interesting, the value was significantly shorter than that obtained in neurons. This difference was curious for us. Then we analyze the difference in neuron and hex cells by focusing the recycling of amplitudes. To analyze the recycling amplitudes, after CAM2TCO labeling 4 hours in neurons, surface amplitude were labeled with tetragen FL as a first pulse, okay? And incubated for 15 minutes. Then the tetragen RX647 was incubated as a second pulse to label exercise dosed. In other words, recycled amplitudes. Western blotting clearly indicates that the recycling amplitudes were observed in cultured neurons. However, any recycled amplitudes were not observed in hex cells. In summary, our two-step labeling reveals the lifetime of amplitude is significantly different between neuron and hex cells. And one of the reasons is that the hex cell lacks the recycling process of amplitudes. As the next directions, we are now analyzing recycling process of amplitude in more detail by using the two-step method, by modifying this method. In this case, after surface labeling, the cells were fixed, okay, surface labeling after fixed. Then they perform the second step labeling targeted for intercellular component. As shown here, the intercellular amplitude was selectively visualized as a different color in hex cells. And some component after internalized has been visualized with a different color. So we think that this method would be used to analyze the recycling process of amplitude in the future. Okay, I would like to move another topic, chemogenetics of cell surface receptors. One of the frontier of neuroscience is understanding the neural network, as you know. This new direction has been achieved by developing technique for activation of target cells such as optogenetics and chemogenetics, maybe another magnetogenetics. Here we focus on chemogenetic approaches. The representative is DRET technique. In this technique, muscarinic acetylcholine receptor is mutated to response to the designed ligand CNO. This receptor lacks affinity to the endogenous ligand acetylcholine. Thus, expression of the designer receptor in the target cell allows conducting downstream G protein signals by designed ligand. One of the advantages of the chemogenetics is controlling the cellular function even in the deep region of brain. Actually, after expression of direct protein using adenovirus virus in the target cells, oral administration of CNO controls the target G protein signal in a cell type specific manner in non-invasive fashion, even in large animals. DRET technique is undoubtedly powerful for controlling nerve activity by chemicals. However, direct protein lack original ligand binding, and the direct protein are overexpressed in most cases. Thus, the ligand-induced signals are largely different from that induced by endogenous ligand receptor interaction. Thus, it is difficult to apply this method for analyze the physiological role of the endogenous receptors. We think that the next direction of chemogenetics would be molecular target to chemogenetics, in which the receptor of endogenous receptor should be controlled in a cell type specific manner, like here. In this direction, 2 points are required. First is selective activation of target receptor by designed ligand by introducing mutation in the target protein in a cell type specific manner. Another important point is original receptor function should not be affected by the mutation. Here we focus on metabolic glutamate receptors mGlu1 as the target proteins which belong to class C GPCRs. mGlu1 is highly expressed in the cerebellum, which has essential role for motor learning. mGlu1 has composed of extracellular ligand bind domain and transferrin regions. Structural analysis reveals that the glutamate binding to the ligand bind domain induced domain closure of the ligand domain and the structure change, this structure change transmitted to the transferrin region for activation of the receptors. We focus on the structure change. And our strategy is introduction of histidine mutation on both upper and lower lips of the domain. And method coordination to the mutated histidine residue would induce domain closure. We expected that if the structure change are comparable to that induced by glutamate binding, the method coordination can activate the mutant receptor selectively. Of note, this mutation site does not join the glutamate binding. Thus, original receptor function would not be affected by the mutations. Based on the structure information of the ligand bind domain, mutation site was selected where distance between upper lips and lower lips become close as the glutamate binding. We designed this double histidine mutant, and functional assay was examined by calcium imaging. We first examined glutamate-induced response in each mutant, and original cell function are completely retained in the 5 mutant. Next, we examined effect of metal ion or metal complex for chemogenetic expression of these mutants. RAF screening identifies the metal complex, pyruvate complex, which activates 3 of the mutant. Among them, we selected a mutant. It is highlighted. As shown here, after addition of the pyruvate complex in the culture medium, prominent calcium response was observed in the cells transfected with the W mutant, but not with the wild-type mGlu1. In our strategy, histidine residue are required on both upper and lower lip, as I mentioned. To confirm our concept, we prepared single mutant, okay? And we expect that this mutant failed to be activated by pyruvate complex. As shown here, the A598 mutant failed to be activated as we expected. However, N264H mutant was activated by pyruvate complex, although the mutant lacked histidine residue on the upper side. We searched other coordinating histidine on the upper side, and we revealed that point mutagenesis reveals that the endogenous H55 coordinate pyruvate complex. In other words, point mutagenesis of the N264H is sufficient for chemogenetic activation. Those dependency of pyruvate complex clearly indicates that the single mutant was selectively activated by micromolar of pyruvate complex. Importantly, glutamate-induced response were not affected by the mutation as we expected. So glutamate response is completely kept even in the mutant. Now we obtained the glutamate mutant and ligand pair, pyruvate complex, or chemogenetic activation. Prior to apply this method to neurons, we examined toxicity of the metal complex to neuron-like PC12 cells. As shown here, the neurite outgrowth of PC12 cells were not affected by the pyruvate complex. However, the cell growth was suppressed by high concentration of pyruvate complex. In fact, we speculated this possibility, cell toxicity, because the chemical structure of pyruvate complex looks like cisplatin. So the inhibition of cell growth would be suppressed by DNA binding of this complex. In other words, we expect that the decrease of the cell permeability of the metal complex improves the cell toxicity. Then we designed 4 hydrophilic pyruvate complex having hydroxy or sulfonate group to inhibit the penetration of the cells of the complex. Among them, more substituted complex successfully activates the mGlu1 mutant. Regarding the cell toxicity, sulfonated version of the complex termed pyrazine sulfo-BP does not affect the cell toxicity even in the high concentration. Thus, we utilize pyrazine sulfo-BP for chemogenetic activation of mGlu1 in neurons. To chemogenetic activation of mGlu1, we prepared knock-in mouse having N6OH mutation by CRISPR-Cas9 technique. In the mutant, not only expression level of mGlu1, but also the function of mGlu1 were comparable to those of wild-type mice. Then we examined chemogenetic activation of mGlu1 in neurons. Here we focus on cerebral LTD. Previous study revealed that activation of mGlu1 in Purkinje cells by DHPG, a selective ligand, induced sustained decrease of EPSP, which is mediated by cation influx via amplifier, which is known as chemical LTD. Then we applied pyrazine sulfo-BP in the current chronic mode in Purkinje cells in acutely prepared cerebral slice, which successfully induced sustained decrease of EPSC. This indicates that our system can be applied for chemogenetic activation of mGlu1 endogenously expressed in neurons. We also succeeded to apply this method for cell type specific activation of mGlu1 using AV in cerebral slices. Please see our paper for the details. Okay, one of the characteristic features of our method is this method can be applied to other receptor family showing the same structure change, such as class GPCRs, other GPCRs, and ion channel type glutamate receptors. So the current method would be one of the useful tools for molecular targeted chemogenetics. In my talk, I reported recent progress for chemical labeling and chemogenetic of glutamate receptors. Now we are developing other method which can be applied in vivo for understanding physiological role of glutamate receptor and other receptor in not only neuron but also in non-neuronal cells, glial cells. Finally, I would like to thank many collaborators, especially for my previous boss, Professor Hamachi, and current laboratory member, and other collaborators such as Professor Yuzaki and Kakigawa in Keio University for electrophysiological assay in neurons. And I also thank Dr. Takahashi and Mizuno in Tsukuba University for making knock-in mouse. Thank you for your kind attention.
Thank you so much, Higeki. Now we have a couple of minutes for questions. We're running late because of the delays that we had at the beginning with technical issues, but if there is any question, either from the audience or the rest of the panelists. And if not, we move the questions, the Q&A to the panelists, to the panel at the end. But Rafa, you have a question, so go ahead.
Yes, this was exquisite, Shigeki. Congratulations.
Yes, thank you very much.
For the chemogenetics part, why do you need the metal? Because it's probably the bipyridines that are doing the binding. No? So couldn't you just do it with an organic compound without any metal coordination? I don't understand why the metal is critical here.
Okay. In the metal coordination, we can control the angle of the binding. Okay, this is good for fix the conformation of the protein structures. Especially, can I share my slide?
Yeah.
Okay, and pyridine bipyridine complex is very useful because two coordination site is already fixed with this ligand. So only two histidine can bind to the pyridine bipyridine complex, and this angle can be fixed to 120 degrees. So we can manipulate the protein structure precisely by using this complex.
So it's more rigid. It's more rigid, the angle, and then you change the rest.
Yeah, in the small molecule, it is surely another possibility is that covalent bond formation by small molecules. However, in this case, the bond is non-covalent. Irreversible direction. However, the metal in the case of metal coordination, this is a reversible bond formation. So we can wash out the metal complex after medium exchange. This step is reversible. So we can control the activity.
Excellent. So if you don't mind, we could pass the word to Xiaoji Duan. He is an associate. Well, first of all, thank you so much for this excellent talk and for answering the questions, Shigeki. And so I was introducing Xiaoji Duan. Xiaoji is an associate professor in the College of Future Technology in Peking University in China. And today she will be talking about nano-enabled brain-wide neural interface, which is a dream to us. So I'm very, very excited and very looking forward to hear what you have to tell us. So without further ado, the conference is yours. Thank you. You are muted. Sorry.
All right. Okay. Sorry.
Now.
Thank you for the introduction. My name is Xiaojie Duan from Department of Biomedical Engineering, College of Future Technology at Peking University. It's a great honor to be here and share some of our recent progress in the field of nano-enabled brain-wide neural interfacing. Okay. Sorry. So we know that most of our behaviors are not simply the result of activity in small sections of the brain, but rather the coordinated efforts of neural activity across a brain-wide scale. So let's take the visual perception as an example. When we see something, information from our retinas gets sent to the primary visual cortex. There it is combined with inputs from other brain regions responsible for object recognition, motion detection, and visual attention. It's this synchronized and coordinated activity across these different networks that allows us to make sense of what we are looking at. From this perspective, in order to understand the neural processes that drive behavior, it's important to study the activity of the brain on a brain-wide scale. So we have several techniques to map neural activity across brain. Some notable methods include electroencephalogram, EEG, an MEG, that is magnetoencephalography, and functional magnetic resonance imaging and functional near-infrared spectroscopy. These techniques allow us to observe and record brain activity across a wide spatial scale without invasive procedures. However, they do have their limitations when it comes to spatial temporal resolution and signal bandwidth. Additionally, it is worth noting that fMRI and fNIR rely on blood oxygenation level dependent, that is both signals, through neurovascular coupling, which indirectly reflect neural activity. So in order to explore alternative techniques for recording neural activity throughout the brain, our team has been focusing on two major directions. First, we are utilizing advanced microfabrication methods and incorporating nanomaterials to develop large-scale implantable electrode arrays or probes. This innovative approach is coupled with our commitment to minimizing the invasiveness typically associated with such electrode arrays. This approach allows us to achieve extensive coverage with high spatial temporal resolution with recording brain activity. And the second, we are working on combining different techniques, such as electrophysiological recording or stimulation with magnetic resonance imaging. This integrative approach capitalizes on MRI's remarkable ability for whole brain structural and functional mapping. By bringing these techniques together, we are expanding the application possibilities of MRI for studying the intricacies of the brain. So now, let's delve into the first direction we are working on. Our focus here lies on intracranial electrocorticograms techniques. ECOGs allow for the direct measurement of neuroelectrical activity by employing a planar electrode array placed on the surface of the cortex. By utilizing a large area electrode array, we can easily expand the coverage of our recordings. The proximity between the signal source and the detector in ECOGs results in outstanding signal quality, achieving remarkable spatial temporal resolution as micrometer and millisecond respectively. Additionally, ECOGs have a very high bandwidth up to 500 Hz. However, it is important to note that ECOG recordings need a craniotomy, and the subdural recordings require an additional durotomy to expose the cerebral surface of the brain. These surgical procedures involved, particularly for large area ECOG arrays, bring forth various operative and post-operative risks, including infections, inflammatory responses, as well as the potential for brain hematoma or edema. Consequently, the invasiveness and associated surgical risks present considerable challenges when it comes to implanting large area ECOG arrays for recording brain-wide neural activity. In addition, apart from the surgical risks, the long-term implantation of ECOG arrays on the brain surface gives rise to additional concerns. You can see here, there are actually intricate connections and crucial mass exchange occurring between the skull and the dura mater, as well as between the dura mater and the brain surface. So when a large area ECOG array is chronically present, it can disrupt these vital connections, hindering the proper flow of necessary substances. As a result, complications like inflammation and brain edema can arise, posing further challenges to consider. So in order to overcome these challenges associated with surgery risks, and ensure long-term biocompatibility for large-scale ECOG recording. We drew inspiration from the application of the self-expandable nano stent and developed a unique device called the shape-changing electrode array, or C for short. This device consists of a thin MEA film, only a few micrometers thick, which is coupled with a shape actuator. Before implantation, the C is deliberately deformed into a narrow strip, measuring just a few millimeters in width. This compact form allows for easy insertion into the cortical surface through a very small opening in the skull for epidural recording, or a small slit in the dura mater for subdural recording. Once inserted, the shape actuator comes into play, facilitating the shape transformation of the C. It unfolds and reshapes itself into an ultra-thin sheet. After retracting the shape actuator, a large area conformal interface can form with the cortical surface, thus enabling brain-wide activity mapping with high spatial resolution and bandwidth. We employed several strategies in the design of the C to address the unique challenges and requirements. Firstly, the electrode array incorporates a multi-layer structure utilizing a web-like carbon nanotube conducting film as its conducting material. This is a vital component of the C, as it allows the electrode to maintain its functionality even during the shape transformation process, which involves very extreme strains. The web-like thin film is composed of interconnected thin bundles of single-walled carbon nanotubes, ensuring conductivity even under substantial mechanical deformations. To ensure mechanical compliance and a conformal interface with the brain surface, the array was fabricated on a thin parylene film measuring only 4 micrometers in thickness. Secondly, the shape transformation is facilitated by a nano actuator. As a shape memory alloy, nano exhibits stable austenite and martensite structures at different temperatures. By heating the nano beyond its transition temperatures, it undergoes a transformation from the martensite structure to the austenite structure, ultimately returning its original shape and size. Lastly, we integrated a mesh structure within the electrode array film. This design serves 2 important purposes: ensuring proper connection formation and facilitating mass exchange between the skull and dura mater, or between the dura mater and the brain. Through our research, we discovered that this mesh structure is very critical in maintaining a high chronic biocompatibility of the large area ECOG arrays. Using these strategies, we successfully implanted the electrode arrays, which covered nearly the entire hemisphere of the brains of the rat into the epidural space of adult rats through a very small cranial opening, measuring only 0.8 millimeter in width and 2 millimeter in length. Here is a video showing an implantation process of a C into a rat. In this particular case, we thinned the rat's skull to provide better visibility during the deployment of the C. Throughout the entire implantation process, we observed no bleeding. Once we removed the deployed C from the rat's brain, we discovered that the brain region underneath the electrode array remained completely intact. There was no visible damage observed on the brain tissue or the blood vessels, as illustrated here. This serves as evidence that the implantation of the Cs did not cause any acute damage to the brain. The strategy of utilizing Cs for minimally invasive intracranial brain activity mapping can also be extended to large animal models. In our work, to minimize the signal attenuation caused by the thick dura mater, Cs were implanted subdurally in the large animal application. The image displayed here captures the deployment of a C in the subdural space of a beagle brain. The C used in this case measured 20 millimeter by 15 millimeter in size. it was inserted into the subdural space through a dural slit about 6 millimeter in length without removing any part of the dural matter. Similar to our previous findings, we observed no bleeding during the entire implantation process. Both in the rat and the beagle cases, the Cs adhered tightly to the brain surface, forming a very conformal interface. Crucially, after the Cs underwent a round of shape transformation, only a very small fraction of channels exhibited a slight increase in impedance, and the majority of channels remained within the required range for successful neural recording, indicating the preservation of their functionality throughout the shape transformation process. To assess the impact of the C implantation process on the brain, we conducted MRI studies. The T2 structural scans revealed no discernible abnormalities in the brain's anatomical features or shape following the implantation. Additionally, the T2-weighted dynamic contrast-enhanced MRI showed no noticeable irritation to the blood vessels in the dural matter or brain resulting from the implantation process. Furthermore, there's no time to explain the details, but the histology studies show that in the acute stage, the C implants elicit a very slight inflammatory response. However, this inflammatory response diminished quickly over time. These results confirm both the minimally invasive nature of the C implantation process and also the excellent long-term biocompatibility of Cs. With Cs implanted through this minimally invasive procedure, we mapped high-quality microECOG activities from rodent brains during seizures and also from canine brains during the emergence period. Significantly, the Cs not only recorded the low-frequency components accessible through non-invasive techniques, but also captured high-quality, high-frequency oscillations reaching frequencies of up to 500 Hertz. These recordings revealed the spatial temporal organization of different brain states with high resolution and bandwidth that cannot be achieved using existing non-invasive techniques. Next, I will proceed with the second part of my presentation, which focuses on the development of MRI-compatible electrodes for simultaneous deeper brain stimulation and fMRI studies. DBS has become a widely used treatment for movement disorders like Parkinson's disease, which also showing promise as a strategy for managing treatment-resistant depression, TRD. Despite its proven efficacy, the underlying mechanism of DBS remain elusive. Functional MRI represents as a powerful tool for mapping brain activity on a whole brain scale. By combining DBS with fMRI in a simultaneous manner, we can gain invaluable insights into the modulatory effects and the therapeutic mechanism of DBS in a wide range of neurological disorders. This exciting avenue could bring us closer to unlocking the full potential of DBS. One major hurdle in DBS fMR studies stems from the considerable image artifact produced by traditional materials used for DBS electrodes during MRI scanning. As an example shown here, a platinum iridium electrode measuring 100 micrometers can generate artifact several millimeters in size. These artifacts create obstacles when attempting to image the brain regions surrounding the electrodes. Consequently, obtaining a complete and unbiased activation pattern of DBS becomes unattainable. We explored the use of graphene for fabricating DBS electrodes. In contrast to platinum, graphite exhibits a significantly closer magnetic susceptibility to water. Our approach involved fabricating electrodes using graphene fibers, which consists of densely stacked and aligned graphene oxide sheets running parallel to the fiber's main axis. These graphene fibers possess high porosity and roughness, resulting in very good charge injection capabilities. In fact, their charge injection capacity surpasses that of most commonly used materials for neurostimulation, and is only slightly lower than that of PDOT. Moreover, our tests have demonstrated that graphene fiber electrodes exhibit superior stability when compared to PDOT electrodes. Importantly, the graphene fiber electrodes showed excellent MRI compatibility. We subjected to a 9.4 Tesla MRI. These electrodes produced an artifact in T2 sequence that was 8 times smaller than that generated by platinum iridium electrodes. Furthermore, in fact, in EPI sequences, the artifact was 4 times smaller, comparable to platinum iridium electrodes in terms of MRI artifact reduction. In fact, not only did the graphene fiber electrodes outperform platinum iridium electrodes, but they also demonstrated smaller artifacts compared to various other electrode materials, including tungsten and carbon fiber. We utilized the MRI-compatible graphene fiber electrodes to investigate the DBS effects targeting the lateral habenule LHB in depressive rat models. The LHB, known as a negative reward center, has shown potential as a valuable DBS target in treating TRD. However, LHB has a very small size, measuring only 3 by 3 by 3 millimeters in the human brain. This compact size presents significant challenges for study with DBS electrodes in human subjects. We implanted graphene fiber electrodes into the LHB of 2 depressive rat models with Takeuto WKY rats, which represents endogenous depression, and rats treated with lipopolysaccharide LPS. These rat models exhibit depressive-like behaviors, including reduced preference for sucrose, increased immobility time in forced swimming test, and decreased locomotor activity in open field test, indicating anhedonia, despair-like behavior, and anxiety-related symptoms, respectively. When we examined the LHB using MRI, we observed the minimal artifact induced by the graphene fiber electrodes, allowing us to easily and accurately verify the correct placement of the implanted electrodes within the LHB. Remarkably, when we delivered the high-frequency stimulation into the LHB using the graphene fiber electrodes, these depressive rats experience an immediate antidepressant effect. This was evident through increased locomotor activity observed in the open field test, the reduced immobility time in the forced swimming test, and also a significant rise in sucrose preference. It is important to note that both the WKY and the LPS treated depressive rats responded positively to LHB DBS, demonstrating a high success rate for this stimulation technique. To gain further insight into the mechanism behind this immediate antidepressant effects of LHB DBS, we conducted fMRI scans on the same groups of WKY and LPS treated rats that was subjected to the behavioral tests. By analyzing the correlation between the antidepressant response and the fMRI signals from various brain regions, we aimed to shed light on the underlying mechanism. We observed the robust, positive, bold responses ipsilaterally along both the direct afferent and the efferent circuitry of LHB. These regions include the medial prefrontal cortex, MPFC, dorsal-raphe nucleus, DRN, ventral tegmental area, VTA, and some other areas. And also we found the activation in some other areas, which is beyond the direct circuitry, such as the most of the activity area are in the direct connections with LHB, but there are 3 regions which are not in the direct circuitry. That is SLEA, cingulate cortex, and retrosplenial cortex, RS. We performed the Pearson correlation analysis to examine the relationship between the behavioral improvements and the levels of both activation, in different brain regions among individual rats. We found the significant correlations between the increase in average speed, the number of entries into the center, and also the distance traveled in the center with the beta values of beta signals, which is a reflection of the activation levels in the septum, DBN, LPA, and some other areas. Notably, all these regions were connected to the medial subregion of the LHB rather than the lateral part. Based on the T2-weighted anatomical images, we delineated the electrode tip location of each rat, which is summarized here. The analysis revealed that rats with DBS sites in the medial LHB and close to the medial habenula, shown as red dots here, exhibited greater improvements in average speed compared to rats with more lateral stimulation sites, particularly those in the lateral LHB and far from the MHB, here shown as the blue dots here. These results suggest that if we want to get a better antidepressant effect, we should stimulate more lateral part of LHB instead of the lateral part. These results, along with the correlation findings mentioned earlier, suggest that the immediate antidepressant effect of DBS primarily originates from the stimulation of the MLHB and also the antidromic and also dromic activation of the circuits connected to the MLHB. We believe that the findings from this research can provide important insights for the development of future DBS therapies. Furthermore, our research highlights that DBS fMRI studies with graphene electrodes can serve as a powerful platform for translational research, investigating the therapeutic mechanism and the modulatory effects of DBS. So in conclusion, our research has focused on 2 main directions in the field of brain-wide neuroactivity recording. Firstly, we have utilized advanced microfabrication techniques to develop large-scale electrode arrays, enabling us to achieve high spatial temporal resolution in our recordings. Secondly, we have explored the combination of other modalities such as electrophysiology with MRI to expand the potential applications of MRI in DBS studies. It is important to highlight the critical roles played by microfabrication and nanomaterials in realizing these advancements. These technologies have been instrumental in pushing the boundaries of our research. With that, I would like to end my talk, and I want to thank my dedicated students and collaborators for their invaluable contributions to this work. And I also would like to extend my appreciation on the funding agencies that have supported the research. Thank you again for the opportunity and the invitation. It has been a privilege to share our work with you, and I'm grateful for your interest and support.
Thank you so much for this amazing talk. So we have a minute or two for a couple brief questions, if there are any. We're running late with the schedule in general, but so if there are no questions, I will save mine for the panel discussion so that we have more time to discuss between all of them. All right?
Okay. All right. Thank you.
Well, thank you so much again. And yeah. Okay, there is a question in the Q&A, but we will save it for the panel discussion because it applies to some other speakers. So with this, it is my pleasure to introduce to all of you to the last speaker of this first session. His name is Michael Krieg, and he is a group leader at ICFO, where he is a professor, if I'm not mistaken. And so he's presenting today a talk with a very captivating title. Well, you can see it already, but the title is Neural Engineering with Photons as Synaptic Transmitters. So, Michael, the room is yours, and tell us about photons as synaptic transmitters, please. So much.
Thank you very much. So I trust you can see my talk and hear me well. So at first, I want to thank the organizers as well for inviting us and giving us the opportunity to present our most recent effort to engineer synthetic neural networks with photons as synaptic transmitters. So my name is Michael Krieg, and I'm a fairly young group leader from ICFO, the Photonic Science Institute in Barcelona, which is on the northeastern part of Spain. This is our institute. And it's not so close, not so far away from Dipsy in the Basque Country, actually. And my group is interested in works in the domain of mechanobiology and optogenetics. And in the mechanobiology part, we are very interested in basic science and the fundamental principles of how cells and animals measure mechanical forces during mechanical transduction, sensory mechanical transduction, for example, the sense of touch and proprioception. So we've heard already in the first talk about the transducing molecules, the ion channels, and we're specifically interested in how these ion channels actually receive the stimulating input. To answer these questions, we developed a mechanical toolbox that spans several orders of magnitude to measure and apply mechanical forces on the level of single molecules in living animals using genetically encoded tension sensors or optical tweezers, but also MEMS and microfluidic devices to apply forces to entire animals and tissues. So at the same time, we can then also read out the deformation applied by these forces and over, again, several orders of magnitude. Our overarching goal with these methods is, of course, to understand not only fundamental principles, but also pathological situations that arise due to, for example, a perturbation of the mechanical properties of neurons and the way how they sense external forces. So in contrast to our other speakers, we apply most of our techniques and our questions to genetic model organisms such as C. elegans, the worm, Drosophila, and zebrafish. But the purpose of today's presentation is slightly different, as we are interested in the engineering of neural networks using novel techniques on neurotechnological diseases. And I don't need to introduce to this audience that the synapses in the brain and neural networks are really the core units where computation happens, and is central for our, let's say, intellectual complexity. So the human brain has a sheer number, uncountable number of them, and that perform all sorts of tasks. And it's quite obvious and understood that a defective neurotransmission leads to a variety of neurological disorders. But it has been recognized early on that actually external interventions through a variety of means, for example, magnetic neurostimulation, electrical stimulation, pharmacological interventions could alleviate potentially some of these diseases and mitigate the burden on the patient. And more recently, also optical interventions by the use of light has been added to this toolbox, and this is going to be the topic of our talk. Why light? In several advantages are conceivable. For example, I mean, it penetrates biological tissues it fairly non-invasively, unless it's in encased by a skull, and it comes at various frequencies and wavelength that allows us to use different receptors, which is one of the core advantages as biological systems evolved specific receptors to respond to light, for example, the ones in our eyes or the ones that we're talking about in this talk. So the idea to use light for stimulating neural activity is quite old. It's been proposed in the late 70s by Francis Crick to use light to activate neurons. But still, it took a few decades until the first application or demonstration was available, which used a heterologous system, orthogonal system in hippocampal cells to activate neurons using 3 or more different enzymatic actuators, which was still quite complicated to implement, to make it really a wide applicable method. But then it happened really fast, as the first demonstration that a algal ion channel could actually be transplanted into heterologous cells and elicit currents up and stimulation of the light. And some of these ion channels are expressed in algae that mediate phototaxic and photophobic behaviors, not necessarily the algae that you see in this video here, but similar ones. And then it happened really fast that these ion channels were also expressed in heterologous neurons and animals to show that these animals change their behavior up in light stimulation. And how that works is that this ion channel can be expressed in any cell type in the neuron, and it can then, based on the absorption of a photon, open its conductivity for monovalent, divalent cations, or if they're anionic, also to anions, to depolarize or hyperpolarize the membrane potential. So this is a really promising strategy in neuroscience, and but there's one obvious challenge that has been already talked about is, and our brain is normally quite dark, so there's no light in the brain, and the central question by actually implementing or adapting this technology into a skull, an animal with an opaque skin, is how do we get the light inside, delivered to the region of interest. Conventionally, this is achieved using light fibers, or so-called optrodes. And here in this picture is an iconograph, an iconic picture with the mouse implanted with an optrode that delivers the light into the region of interest that express the light-gated ion channel. An experiment like these enable studies like the one in this video here, where the mouse is transgenic for a generodopsin in the amygdala, a center for prey hunting and exciting aggression, and the mouse is seemingly bucked by this toy. But then, when the researcher switches on the light, it is delivered to the area, the mouse starts attacking and changes its behavior quite dramatically. So these experiments are very insightful and very informative to decipher neuronal circuits, but they all rely on the delivery of the light through an optrode or similar devices. But nature actually implemented, or basically delivers already, ways to potentially replace the optrode using enzymes that produce light based on enzymatic reactions. And one of these implementations you see in this little video here, which are not bioluminescent dolphins, but dolphins swimming through dinoflagellates, which itself are fluorescent and emit photons based through as a calcium sensitive enzyme that is activated by mechanical shear forces as the dolphins flow through water. So our approach to that field is to take advantage of these enzymes and encode them in the presynaptic compartment of a neuronal circuit that then would emit photons that are sensed and activated in postsynaptic compartments through classical light-gated ion channels. We implement such a system, or we work towards such a system using the small genetic model organism C. elegans that you can see crawling through that window here. It's transparent, which is very good for our purposes to develop and and prove such a concept. So we can also read out neuron activity based on calcium indicators. And here's a video where the whole brain has been labeled with a GCAM, and each of these fluctuations signal a neuron activity event, in that case, due to oxygen levels and so on. Moreover, C. elegans has a complete known wiring diagram with synaptic connections that are very well understood, and circuits that are also functionally beginning to be characterized. For our purposes, we take advantage of a mechanosensitive avoidance circuit that sits in the nose of the animal. So it's basically one of the neurons that innervates the nose tip, and it senses mechanical touch through connections with two interneurons called AVA and AIB that then elicit a reversal behavior. So the numbers here represent representative numbers for the amount of synaptic connections between these individual neurons. So just to provide the level of details that people think about this neuronal circuit. This is the behavior here, what is mediated by these circuits. So as soon as the animal navigates into an obstacle, and it reverses its crawling direction to eventually avoid this obstacle. The circuit depends on glutamate signaling. So the predominant neurotransmitter is glutamate that then is secreted from ASH and taken up from the interneurons, and deletions of the glutamate in the animal render the system, the organism, completely insensitive to this nose touch, as we can quantify using our pretty crude assay here. We also established a microfluidic device to interrogate the flow of information through the circuit based on a mechanical touch to the nose. And this device is schematically seen here, in which we have a trapping channel where the animal sits, and then a pneumatic actuator that then delivers a mechanical insult to the nose, while we can read out mechanical behavior, neural activity. Right, so this is obviously the nose, the diaphragm is driven into the nose, and then we can read out neural activity using calcium imaging. In these following videos down here, I show you two examples of a wild-type animal that expresses R-gecko in AVA, one of the interneurons, and another animal on the right-hand side that is mutant for glutamate signaling. So what you can see is that the left animal, which is wild-type for glutamate, responds to the nose touch, whereas the right one does not respond to the nose touch delivered in this device. We can now repeat this experiment for many different animals and in many different conditions. And in the wild-type situation, we observe a robust neural activity in the sensory neuron, but also in the two interneurons, AVA and AIB. But as soon as we take out glutamate from the animal through specific mutations, then we see that only the sensory neuron ASH responds to the nose touch delivered to the animal, but not the two interneurons. So this is our baseline phenotype now, from which we started to finally replace or recover that chemical, this defective chemical transmission with photons through the bioluminescent reactions. So our aim is to engineer prosthetic synaptic interface with photons instead of chemicals. As I said, we use luciferase to emit the photons, and we use not the one from dinoflagellates, but we use one from shrimps, which is the most powerful luciferase known to date, at least to our knowledge. And we can select and make them more efficient by fusing with the fluorescent protein. It's been described and suggested in Suzuki et al. In 2016. In addition, we make this luciferase sensitive to calcium, which means they emit efficiently more photons in high calcium concentrations that are reminiscent for neural activity. For example, in the situation where ASH is active, we get a higher activity of photon emission. So we wanted to see this live in the worm, but our commercial standard epifluorescent microscopes were unable to deliver images at exposure times that are compatible with the swift biological dynamics that happen during neuronal signaling. So we improved a current luminescent microscope and combined this with deep learning algorithms to eventually record activity in moving animals down to 150 milliseconds. And then we observed, for example, here an animal that is freely moving with a calcium indicator, bioluminescent calcium indicator in the muscles. And what we observe is indeed a higher photon emission at the contracted side of the muscles, where a high calcium concentration occurs due to its activity. So that approach also works in neurons. And so here you can see a few examples. And my student then went on and combined that methodology with light field imaging in order to obtain a whole 3D representation of the scene with a single exposure and bioluminescence, which provides us actually a 3D render, a 3D representation with 200 to 500 millisecond exposure times. And here indeed, we again see the same, where we see higher photon emission at the contracted side, where the muscles signals to the calcium, and the calcium activity follows precisely the curvature of the animal, therefore the muscle activity. So with this, we are quite satisfied to have seen that these luciferases work well in C. elegans. And then we turn to the postsynaptic side and engineered a channel rhodopsin with a high operational light sensitivity into ABA and AIB. We employed a selective genetic strategy that I don't want to go into detail for this talk, but we achieved the single cell specificity in AIB and ABA, which you can see here, for example, the localization of a tag channel rhodopsin at the cell periphery. Functionally, we can then ask, do these animals respond to light? And here's one video in AIB. And yes, when the light switches on, the animal reverses its behavior, reminiscent or like similar to the endogenous behavior that the animal would do in the nose touch assay. So here's the other neuron ABA. So the light is delivered and they will reverse their direction. We then titrated different light levels into these transgenics and observed the light response of these animals. And if you then fit the data, fit the model with the data, we can then abstract to low light densities and then estimate that these animals would still have a sensitivity